Welcome to today's webinar. My name is Carolyn Marks, and I'm a research scientist at Atlas Antibodies, and I will be moderating today's webinar. We are live here in Stockholm at our company headquarters. Before we start, I'm going to go through one practical point. Feel free to use the open chat function and QA functions at the bottom of your screen at any time throughout today's webinar. After the webinar, we will invite you to please fill in a very short survey, which will show up in your browser when the webinar ends. We really appreciate your time and feedback. So the topic of today's webinar is exploring neural lineages with brain organoids and their implications for understanding health and disease. Our guest speaker is Dr. Veronica Pravata. Veronica is a postdoc research fellow in the lab of Professor Silvia Capello at the Biomedical Center of LMU in Munich. She is working on the role of post-translational modifications, or ampylation, in brain development and autism spectrum disorder, using a combination of proteomics and human-derived cerebral organoids. After her, Dr. Eugenia Kutiva, a principal scientist at Atlas Antibodies, will present our portfolio directly related to this research area. We are very much looking forward to having an interactive session with you today, so please feel free to ask questions at any time using the Q&A function at the bottom of your screen. We will gather your questions and answer them after both speakers' presentations, so please ask us and we will happily respond. Finally, we will also be asking you a few poll questions after each presentation to gather your opinion and some experiences on this engaging topic. And now, Veronica, welcome, and you may start your presentation.
All right, thank you so much, Caroline, for the nice introduction. And I hope you can see me and hear me, and you can actually see my pointer. So as Caroline already mentioned today, I will talk about how we explore neuronal lineages using brain organoids. So I will give you an overview of the state of the art in the field in brain organoids and how we actually use brain organoids to understand disease and other type of research questions that we have in our field. Now, as you probably are very well aware, our brain is a mystery. We really do not know that much about it. It's an extremely, we all know, and probably you all already know, it's an extremely complex organ, right? And it controls our body, provides us with sense of self, but we really do not know much about it. There are many questions that we're still trying to tackle, from very complex ones, such as, sorry, my pointer is not working, such as consciousness, but also, okay, sorry, my pointer wasn't working. Also connectivity, neuroplasticity. We don't really understand how to tackle neurodegenerative disorders. And first and foremost, we really do not know what makes us human. So the question that we have in our lab and in many other labs is, how do we build a brain? How brain development works? Because after all, one of the things that we really do not understand is actually even how brain develops. And this is probably given by the fact that brain development is an extremely intricate complex that is given by an extremely heterogeneous cellular complexity and a very well-timed differentiation. We have, as I mentioned, different types of cells that are progenitor cells, such as the apical radial glia, basal radial glia, and intermediate progenitor that give rise to extremely different and complicated and interconnected type of neurons. So we actually have different type of neurons that are arising in a very wide span. So usually we look at we can look at post-conceptional week. Usually this goes from post-conceptional week 6 until post-conceptional week 24, for example. And this is where actually most of our neurons and different type of neurons arises. But also this is actually a very long process. This happens over a span that goes until after actually conception. So what are we looking here? That's a very beginning of neurodevelopment. And there are not just only neurons, right? We also have other different cell types. We have astrocytes, microglia, other interneurons that migrate from very different parts of the brain. And therefore, one of the main questions that we do have as neurodevelopmental biologists is how do we study such complexity and brain development in general? Now, needless to say, there are a lot of amazing models that are recapitulated in the last decades, neurodevelopment, and each of those have amazing strengths and weaknesses. And this is really obvious for any type of model. But what is extremely, I think, complicated and we need to keep in mind is that in brain development, really this cellular heterogeneity and this dynamicity, it extremely varies across species and in human brain development specifically. And it's actually quite complicated to study. So people have tried to use several model systems, so very from more to simple, like in vivo models that are, again, they gave invaluable insights in neuronal development, such as, Drosophila, zebrafish, mouse, primates as well. But obviously, those are extremely complex models. In vivo models takes very often a very long time, and they cannot always recapitulate what happens in human brain development. And because of that, people try to look at in vitro models that can be derived from, for example, from induced pluripotent stem cells that are derived from human. And because of that, like obviously there are different type of cultures, in vitro systems, such as 2D cultures, for example, of neurons. You can have co-cultures of neurons and astrocytes, for example. And you can also have more complicated and slightly 3D models, such as spheroids. However, when you go into in vitro models, those lacks of that cellular heterogeneity, that it's a key component in neurodevelopment. And really because of that, like people have started looking at new models that could, from one side, recapitulate the cellular heterogeneity and complexity of brain development, but on the other side, really encapsulate the human specific changes and peculiarity. And that's where actually brain organoids came useful. Now, just to give you a very quick overview of brain organoids are made. Brain organoids usually derive from stem cells. They can either be derived from embryonic stem cells that usually are taken from blastocyst, but very often people actually take advantage of induced pluripotent stem cells because those can be actually derived from fibroblasts from either humans, primates, mice, or literally virtually any type of animal. And you can actually reprogram them using the Yamanaka factors and having induced pluripotent stem cells. Now, both induced pluripotent stem cells and embryonic stem cells can be used to generate a brain organoids. And once you generate an embryonic body, which will have 3 different germ layers that are part of like a normal development, in the generation of brain organoids, you will specifically develop the ectoderm, so the outer germ layer. And the beauty of the brain organoid is that you could actually recapitulate that cellular complexity that I already previously shown in that is actually the main key component in brain development. Obviously, the time scale is slightly changed. Usually you start having at around day 20, most of your progenitors that will populate the ventricular and subventricular zone of your brain organoids. And around day 60 onwards, you will start populating your cortical plate with your different neuronal layers. And after approximately day 90 and onwards, you will have, for example, astrocytes and more interneurons sometimes, and so on and so forth. Another amazing component of brain organoids is that we can virtually generate pretty much any type of portion of our brain while developing. Obviously, the brain, it's during development, it's an extremely complex, and this is like, I'm stressing it because really this is a key aspect about brain development, and we have different components, and different scientists across the world, once they develop the brain organoids protocols, they try to generate different type of brain organoids that are defined as pattern organoids. So at the really beginning, what people had was kind of a pattern organoids in which you have features of both the cortex, so the dorsal portion of our cortex, but also the ganglionic eminence portion. So you will have actually a mixture of these components. And again, this is like actually a titanic effort from several labs that have been around the world. A few of them, Medellin Lancaster, Sergio Pasca, Jürgen Knoblich, Kwame Ling. So those are all amazing scientists that really like develop these tools. And I really suggest you to go to there are different methods in which we achieve different and similar actually type of organoids. However, another huge strength about brain organoids is that you can really derive very specific subtype of the neocortex. So, for example, you could virtually generate dorsal pattern organoids. So they will actually have features of pretty much just the cortex, but you could also be interested in the ganglionic eminence and therefore generating ventral organoids. We will have features of both the lateral ganglionic eminence, the medial, and the striatum. And you could also go even more in depth into more specific, going, for example, into generation of the striatal organoids that have been also extremely valuable tools, depending obviously on your biological question. At the same time, there have been also other type of organoids, such as the vascular organoids, which give rise to the blood vessels and microglial cells, and those are usually derived from a different germ layer, so that's why usually our organoids missing it. And because of that, people start playing with different type of organoids and assemble them to look at different aspects. So for example, you can have dorsal ventral assembloids in which you literally assemble dorsal and ventral organoids. And you could actually study how inhibitory neurons and interneurons migrate towards the cortex. And at the same time, with the use of vascular assembloid, you could also study how blood vessels and microglia migrates in the cortex, and so study more complex and events. But we're here to understand how we can actually use these brain organoids and what type of question we can ask with organoids. I think we can pretty much divide the type of question that we can ask in three different aspects. So we can look at physiological differences, we can look at disease, and we can obviously look at developmental changes. And what I really want to make clear is with the brain or human brain organs, we can really look at very human-specific type of questions. So just to go at the beginning very briefly of the type of cell types that you can identify, also to show you that famous cellular heterogeneity that I've been talking about. I will give you just a brief overview of the different cell types that you can see within a brain organoid. So those are, for example, brain organoids that have been stained for proliferating progenitor cells. So those areas, those are very condensed type cells are actually generating the ventricular zone, and the outer part will mimic the cortical plate. And you can stain again for very different type of cell type, and really this gives you an idea of the heterogeneity of the cell types that really resemble human brain development. So you will have, for example, phosphohistone 3 of Ki67 positive cells that mimic proliferating cells, but also more mature neurons, such as, for example, mu and inhibitory neurons that are then usually for calretinin, and part of neurons from the cortical plate, such as CDIP2. But you can also go more in depth and more in detail and go more molecular. And you can also stain for different type of markers for different organelles. So those are just a few examples, such as GM130, which is a marker for the Golgi, PDI for the ER, TOM20 for mitochondria, for example, and CD63 for lysosomes. And also, to give an example, you can also look at post-translational modifications. So, for example, the PRO-N6PA probe is a probe that can be by which I usually visualize AMP lesions. So the post-translational modification that I work on. And so this is really to give you an idea of like how in depth you can go within an organoid, and how many different things you can look within an organoid, and how well they can recapitulate brain development. And since they can recapitulate very well brain development, those are actually being invaluable tools for understanding disease. So there are very different neurodevelopmental disorders are very heterogeneous and happen at very different times of these, again, very well-timed differentiation into these different type of cells. And people have been trying to classify them, categorize them according to the defects that can happen, right? There can be defects in proliferation, and those are usually leading to microcephaly. There are defects that can happen in migration, either in interneuron migration, but also in neuronal migration towards the starting from the radial glia. And those usually leads to periventricular heterotopia, for example, polymicrogyra. And also can be obviously later on effects in synaptic activity and synaptic and axon growth. And this usually leads to other diseases such as autism. And so today I will bring you a very example of something, of one of these diseases that has been it would have been impossible, I think, to really appreciate it if not in a human context. And this is given by this L-galestre-BP protein. Is a protein that one of the candidates protein in our lab. And so far was very well known as actually cancer biomarker. And L-galestre-BP is a secreted protein. We identified very recently in individuals that show changes. They show usually periventricular heterotopia, which is a cortical malformation, and autism spectrum disorder and epilepsy. What was actually astonishing about these patients is that many of these patients show altered gyrification. And the gyrification, so I will show you the example of one of the patients that we have we've been working on in our lab. And the gyrification is basically the folding of our brain, right? And these patients really show changes in the gyrification, the so-called depth and thickness of their cortex. And this would have been very difficult to study, for example, in mice model, because mice are lissencephalic models. So their brain is completely smooth, and gyrification is something that happened actually later on in evolution. And because of that, we decided to study how L-galestre-BP is altering brain development using brain organoids. Doing some transcriptomic analysis, we identified that L-galestre-BP is highly expressed in radial glia, and in particular is expressed in basal progenitors, including intermediate progenitors and basal radial glia. Those are cells that are usually generated from the apical radial glia, and are actually enriched in gyrencephalic species like human. And therefore making it an extremely human enriched protein. And because of that, we decided to look at these basal progenitors. One of the markers for it is, for example, HOPEX. And this is an example of a control, so healthy derived brain organoids. And this is how it looks like. Once we looked at brain organoids that were derived from one of the point mutation that we identified in patients, it was very surprising to see that these HOPEX positive cells, or the spatial progenitors, weren't laminating properly. And so changes in L-galS3BP protein had a huge impact in basal progenitor delamination and overall in their function. And so just using cerebral organoids, we were able to really validate and demonstrate that these changes in gerification are given by this alteration in L-galS3BP, which is a human-enriched protein, and it's a basal progenitor-enriched protein that, and again, basal progenitors are highly enriched in humans, so it would have been, again, impossible to study it in other models. And again, this is one very nice example about it, but that's not all we do. We also try to look on an evolutionary point of view, how brain development is different. And studying from an evolutionary point of view, how similarities and differences between hominids, for example, is quite important for understanding genetic, molecular, and cellular basis of human brain specialization. So very recently, many people try to look for single nucleotide variants in DNA from closely archaic hominids, such as, for example, Homo neanderthalensis. And thanks to next generation sequencing, several single nucleotide variants in the DNA of Homo neanderthalensis were identified. And so people start thinking whether these differences, these single point mutations, could somehow explain the differences between, for example, Homo neanderthalensis and Homo sapiens, and how they could, for example, explain differences in very human-specific brain development. And among these single nucleotide variants, there is one that was identified in the protein known as DAXUS1, and in particular in this cadherin 7. So DAXUS1 is a protocadherin that is involved in axon guidance and dendrite arborization during development. And this, again, is one of the proteins that we have been studied for a very long time in our lab. And for us was actually quite surprising because this protein bears an amino acid change between Homo neanderthalensis and Homo sapiens. And this made us really think how changes in the amino acid sequence of DAXUS1 could affect neurodevelopment from a really evolutionary perspective. And because of that, we decided to generate brain organoids of both Homo sapiens, so wild-type DAXUS, and what we call as a Neanderthalized organoid. So we insert the point mutation that was found in DAXUS into human brain organoids. And I will show you a few examples of how this single point mutation in DAXUS could alter brain development. So we look, for example, changes in the progenital zone. So for one marker, a very broad marker is PAX6. And as you can see, the thickness of the ventricular zone of brain organoids from human and Neanderthal is quite different. So we have a reduction in the thickness of the ventricular zone in DAXUS, in the Neanderthalized DAXUS compared to the wild type. And this can be seen at early stages, or around 30 days, in which we have mostly progenitors, and later on when there are actually also more neuronal subtypes that are populating the cortical plate. And also to give you an example of markers for cortical plates, such as CDIP2, we also see difference in the number of CDIP2 positive cells between Neanderthal, where there is a reduction in a number of CDIP2 positive cells compared to the human DAXUS. And since Daxus is quite important in migration, we decided to look at the arborization of the processes. So something that can be done is to look, for example, at double cortin, which is a marker for neuronal processes. And you can try to study, even in a fixed manner, whether the processes are stuck in the ventricular zone. And as you can see here, basically in the Neanderthalized Daxus, we have more processes stuck in the ventricular zone compared to wild type, meaning that also the migration of these neurons is altered. And this is, again, is a really work in progress. And I really this is just to give you an overview of the different, how amazingly, I think, well, brain organoids can really recapitulate the different neuronal lineages that are present in the brain, and specifically during brain development. We obviously just scratch the surface of the strength of brain organoids. Brain organoids have been used for a relatively long time, and we use for many different questions. So again, human evolution, brain development, a more mechanistic question, and they can be, but they're also extremely good tools for many different techniques. So they are very easy to be genetically engineered. And for example, the one that I showed you have been CRISPR-Cas9, have been like modified using CRISPR-Cas9, but organoids can be used in large scale for drug development and genetic screening variants. And about the other amazing things that can be done with organoids that we currently do in our lab is actually electrophysiological studies. So organoids do show electrophysiological properties. And also we can use omics analysis, such as transcriptomic, proteomics, both at the single cell level, spatial omics, to really study characteristics of brain development and hopefully to better understand development. And I would like to leave you with just future directions in terms of like in which direction we are going with brain organoids and brain development in general. I think I talk a lot about how amazing brain organoids are, but we need to keep in mind that it's, again, a model, and such as any model system, it has its limits. So obviously, this is a very good review that really recapitulates the difference between a human developing brain and human brain organoids. And we really need to keep in mind that there are missing and underrepresented cell types, such as, for example, microglia and endothelial cells. And these are key components in brain development. And the direction in which scientists are going in the brain organoids field is to try to overcome this lack of microglia and in particular mature microglia and endothelial cells, so like blood vessels, using brain transplants. So the idea is to generate brain organoids and use literally mice model or rat models as an incubator for the organoids to further develop and mature. And this has been shown to be extremely working quite well. It's still, again, we are very early on in this part of the field, so there are still many questions that have to be answered. So, for example, they're also generated for getting active microglia and study human microglia. That is one of these things that has been extremely difficult to study into this system. And this is, again, just to leave you with an idea of like what else could be done and what is the direction. And with that, I would like to simply thank my lab, in particular Silvia Cappello, which has been an amazing mentor, and she has been really helping me going through my postdoc and people in my lab. And obviously from the evolutionary Daxus One project, Svante Pabo and Stefan Riesenberg, which are the people that identified and are collaborating with us. And with that, I would like to thank you all for your attention, and I'm very happy to take any question later on. Thank you.
Thank you, Veronica, for a beautiful and really insightful presentation. We would now actually like to have a few poll questions related to her talk, and we will save all questions to the end and have time for that. But you can vote on the screen and the results will be displayed immediately after. So we'll start with the first question. What is the primary purpose of using brain organoids in research? 1, to study how the brain works, to develop new medicines, to grow full-sized human brains. To study how the brain works and to develop new medicines. Those are the top 2 I would pick as well. Thank you for your input on that. The next question. What are brain organoids typically derived from? Stem cells, bacteria, or animal brains? Stem cells. Although there are some transplantation studies using animal brains, I will say, that are quite interesting. And our last question for now. How do researchers study brain organoids? Using advanced imaging techniques, through psychic abilities, by performing surgery on the organoids? I'm looking forward to this answer. Yes, somebody actually did submit through psychic abilities. Using advanced imaging techniques is the primary purpose, but also there's different surgical procedures that can be done, and I wouldn't say telepathy or psychic abilities, but definitely they respond to retinal stimulation and calcium currents. So thank you for your valuable input on these polls. We will now return for a final presentation from Dr. Eugenia Kutiva. Eugenia, welcome, and you may begin your presentation.
Yes, hello. Thank you, Caroline. Well, all right. Well, first of all, thank you really, Veronica, for this excellent talk. What a fantastic studies you are doing, really inspiring. And beautiful images. We always love to see that. But in the second part of this webinar, I will focus more on Atlas antibodies and our product and how can they be used for the neuroscience research. We have a really large catalog of primary antibodies that includes both polyclonal and monoclonal antibodies, targeting virtually all human proteins. So together, these antibodies are covering a large number of research areas, and not least the neuroscience, of course. And they can be used in multiple applications, such as immunochemistry, Western blot, and immunofluorescence, for example, in the cell lines. Although the antibodies are developed primarily against human epitopes, you can also use them in other species, especially in rodents, which can be used as the model when we are studying the human pathology and biology and development, for example. And last but not the least, we do offer antibodies of different clonality, so both rapid polyclonals and mouse monoclonals, and different isotypes, which actually can help you and enable for easily performing different multiplexing studies. And I will demonstrate some of such examples a bit later. At Atlas Antibodies, we also pay particular attention to the antibody validation and thorough antibody validation. And we use both the standard and the enhanced approach when we do develop our products. First of all, already at the time of the development and antigen design, we carefully design our antigens aiming for the most specific and immunogenic part of the proteins. We also validate them in different applications, as I already mentioned earlier. So Western blot, immunohistochemistry, and immunofluorescence are the core applications. But we also add an extra layer of the validation, such as genetic, orthogonal, or independent antibody validation to really make sure that we can provide our customers the most reliable results. Neuroscience, the use of antibodies in neuroscience. What I was saying is that we do have a really large catalog of antibodies for neuroscience research. And just to give some examples, we do have antibodies targeting, for example, different cell types in the nervous system, cortical markers, neurodevelopmental markers, but also antibodies that can help you to study neurosystem and brain pathology, not least glioma markers for looking at the brain tumors, or for example, multiple sclerosis markers that you can find in our catalog. So as we have just heard in the fantastic talk by Veronica, the nervous system is actually a really complex organ that is built by many different types of cells. And to be able to identify these cells that are present and where the particular cells are located, how they're communicating and interacting with each other, that can help us to understand both the normal development of the brain, but also what may go wrong, for example, during development or different diseases of the nervous system. And this is just an example of the cortical development and the presence of the different markers that can help you to define different neurons in the cerebral cortex. And these markers, they can be used for identification of different cell types during development and also in the adult brain. And they can be visualized, for example, by using multiplexing immunofluorescence immunohistochemistry by combining primary antibodies raised, for example, against rabbit and mice. And mice do have, for example, different IgG classes. So you can easily combine, for example, G1 and G2 or 2B classes to create multiplexed staining of the proteins that you're interested in. And here is just an example you can see to the right of the laminar structure of the cerebral cortex in red, where you can see the upper neuron labels, upper neural layers labeled with the marker against HOOX1 that you also can see here during development present in the brain. Layer 4 is marked by a calcium binding protein antibody. And finally, the deep layers 5 and 6 are stained by FOXP2. And here we combine both polyclonal antibody and 2 different types of monoclonal antibodies. Just to show you that these antibodies can also be used successfully in studying embryonic tissues. And here we did apply them in the mouse brain. And you can see, for example, the progenitor marker PAX6 and the TBR2 that are present already at the embryonic day 11 for the mouse development, and how the cortical plate continues developing during the embryonic day 14. Here is another example, just to show the development of the neurons using different immature neuron markers, such as tubulin beta 3, double cortin, and for example, TBR1. And here in the lower pane, you can see how the neurons are appearing at around embryonic stage 11, and how the how the cortical plate is continuing growing. And finally, the expression in the adult brain. So you can find more information on these and the other antibodies for neuroscience research on our website. For example, in our recently published white paper on neuronal lineage markers. And with that, I would like to thank you for your attention. And I think I will get over to Caroline.
Thank you, Eugenia. That was a beautiful presentation. It's nice to see our markers in action. They are gorgeous. Now I will invite you to answer 2 final poll questions. So how are primary antibodies used in brain research? To label specific proteins or molecules of interest, to create brain organoids, or to study neural networks in real time? Primarily to label specific proteins or molecules of interest. And our last questions. What is the advantage of using primary antibodies in brain research? They allow for precise detection of specific targets, They enhance brain organoid growth, or they eliminate the need for advanced imaging techniques. Unanimous, they allow for precise dissection of specific targets. This concludes our polls for today. Thank you very much for your participation. Your input is truly invaluable. And now, Veronica, Eugenia, and myself will address your questions. If you have any additional questions, please don't hesitate to add them in the Q&A in the bottom of the slide. So we can start with the first question. I think this is probably geared towards you, Veronica. Where do you get the
I guess it's iPSCs.
IPS, yeah. Where do you get the IPS cells from, and what differentiation media do you use?
Yeah, so iPSCs usually are either commercially available, in particular control lines. It depends then if you are looking for specific like disease, for example, models. Obviously, there are two ways to get them. One way is to, if you have your control lines in your lab, is to use CRISPR-Cas9 to engineer the mutation that has been identified in a specific disease or wherever you're really studying. Otherwise, there are some databases and biobanks in which you can get the iPSCs from patients, and they have been obviously validated. They all have like all the permissions to work with them. Assuming that you have the permission in GeoDed, these MTAs to get working with iPSCs. The way we differentiate them, it really, there are different medias that you use during the time. The main idea behind that is that you really, at the beginning, you generate the 3 germs layers, I was saying, so ectoderm, mesoderm, and endoderm, but then you really focus on the ectoderm. So all the media that you use, again, there are several, but the main idea is that these work towards expanding the neuroepithelia at the beginning, so the ectoderm that will become neuroepithelia, and then differentiate these neuroepithelia into neuronal types. So usually it's a combination of different factors and molecules. Again, all these media and composition are extremely available and well known in many publications. And it really depends, again, obviously, if you want to pattern them, you will use specific molecules to pattern then your organoids that you will add to this media.
Excellent. We can move on to the next question. A limitation of brain organoids, as you mentioned, is that they do not contain all the cell types, in particular vascular cells and immune cells. How can these limitations affect the overall results when you use them as disease models?
Yeah, that's a very good question. It really depends, obviously, on the type of disease you're looking at, and that's why I think one powerful way of overcome that it's if you, for example, know that microglia has an important impact in a specific disease. Nowadays, there are these assembloids that you can generate. So you might want, for example, to use, yes, your cortical organoids, but add, for example, these vascularized organoids that do have microglia in blood vessels. I think the quick answer is it really ends up to what's your biological question, and then you try to adjust your model in that direction. But obviously, it's important to keep in mind that there are these limitations. And another obviously option is to nowadays transplanting the organoids in mice, for example, as I was showing in the end of the talk.
The next question is, hello, I'm interested in this subject, but I have no experience in the field. I would like to know how we can correlate the development of the human brain with the development of an organoid brain. The timing is not the same.
Yeah, that's absolutely right. Fortunately for us, there have been, again, many people have been done a titanic job towards trying to figure it out how we can compare different timings, because obviously a real human brain takes way longer than a brain, the brain organoid, thank goodness. So the idea is that many people have been doing single cell RNA sequencing analysis, for example, and they try to really age match the timing. So through single cell analysis, you can look at transcriptomic patterns and really try to figure it out, the matching and match the age, the time. And this is something that nowadays it's relatively well known. So again, there are papers in which you can see that, and you can really try to correlate these. But yeah.
There are a number of also like calcium imaging studies and electrophysiology, just like you mentioned, they're doing in your lab, Veronica, where they'll actually cellular phenotype
Absolutely
be able to clock the timing throughout development, which is, I think, quite crucial as the markers then are expressed at different stages. So it really resembles, I think, quite nicely either models of disease or the development of the human brain.
Yeah, absolutely.
The next question, could you comment on the heterogeneity and reproducibility of brain organoids between different iPSC cell batches?
Well, that's again a good question, and I think it's a question that really comes up very often, because one thing that obviously people think is always how heterogeneous, and really this ends up to the fact that we know how to make brain organoids, but we don't know fully like how brain development works. So how do you guide perfectly something into a mature organoid? I think nowadays is becoming more and more reproducible, because we really are getting a grasp of how to work with brain organoids. In general, yes, there can be differences, in particular depending the different background, right? Every cell lines, every iPSCs will have a genetic background that is different, but this can also be a power, because obviously it means that like it's like when you look at population study in a way, obviously in a very simplified way, because each genetic background will give rise to then variability, but this variability can be used also to understanding what are common mechanism and patient specific then, for example, changes. So in general, like nowadays we do not have huge differences, in particular in controls. So when you differentiate them, they more or less overall are behaving fine as far as they are being karyotyped. You need to constantly basically karyotype your cells because iPSCs are very prone to chromosomal aberrations. I think there is a lot of like quality control that has to be done. You need to make sure that they are actually pluripotent. Sometimes they tend to be not fully pluripotent. So a lot of quality control at the beginning really helps you and saves you a lot of time and gives you the higher reproducibility overall.
That's perfect. Is Atlas Antibodies a supplier or a manufacturer of antibodies? I can answer that one. We are distinctively a manufacturer of antibodies. We do have suppliers and distributors, but we are the manufacturer. And we make and QC everything ourselves with high validation standards. There's a lot of people very interested in your contact information, Veronica, which is on the screen as we speak. We have another question. Do you sell carrier-free antibodies suitable for FFP? Our IMC conjugation kits aren't compatible with BSA. I guess that goes to us. So we do have different bulk requests, and we do make different antibodies in different formats for specific customers. So if you do write to our customer service and request or ask, we will do everything we can, and absolutely we have in the past made carrier-free and glycerol-free antibodies and in bulk quantities. So please submit your request to the message or the contact at the bottom of the screen. I'm reading a work that generated a whole mouse brain of E11. I don't think that's a question, so we'll move to the next. What are the main challenges in the preparation and analysis of brain organoids?
Well, I think in general, like the main, it really boils down to what type of protocol you're using. For example, if you're using assembloids, you really, while you're preparing them, it's to really technical, making sure that you're putting them in the right position. So they're really technical things that you usually learn while you're doing them, and the more you do, the better. In terms of analysis of brain organoids, I don't think they're that terribly different compared to other models. I think the only main difference is, yes, they recapitulate brain development, but the whole organoid is somehow, sometimes, depending on the protocol, can be more heterogeneous than other. So you need to be very sure that what you're looking at is, for example, a ventricle, and it's either a dorsal derived ventricle rather than a ventral if you're looking at a pattern. So you really need to make sure, again, there are quality controls that we do usually. You don't just batch produce a full set of organoids and you blindly look at them. There are quality controls that you can follow during the development. And during the analysis, yes, same thing. There are a few things that you need to look. But in general, yes, I think the only main difference is that there might be areas in the brain organoids that are not fully differentiated, for example. And so those might be areas that you don't want to really look at, and you need to, for example, filter for it.
Is it possible to create fusion brain organoids with the blood-brain barrier? If yes, how could we do that?
Okay, so I know there are people working on it. I know that there are, again, people that, yes, are working on it. I think the closest thing that so far we have, it's obviously the generation of these vascularized organoids. I don't know if I don't think they fully recapitulate the blood-brain barrier, obviously. But again, I know that there are, because obviously it's such an interesting idea of like having a model for the blood-brain barrier and trying to understand what can permeate. And again, yes, there are people working on it, but I don't think we have done much yet, or at least I'm not too aware, because it's a bit far away from what I actually studied, if it's actually something that it's working efficiently.
Yes, I agree. But I would say patience is what's necessary now. But I know that there are some labs coming out with this in northern Sweden. So soon. Do you think that the brain organoid can be a good model to study aging disease by comparing the development of a brain organoid to the human brain?
So I know that there are people that use brain organoids for, or they have been tried to use brain organoids for studying aging. I think the important thing is, in general, you can use, I really feel like you really can use whatever model you like as far as you're aware of its limits. And I think brain organoids so far, they've been an amazingly invaluable model for very early developmental stages. I think in that case, they recapitulate very, very well human brain development. The further you go, the more they struggle, I think, to maturate to a point. So they really reach a limit, and that's why people, they've been moving in this transplantation, because at that point, once they transplant them, they really see that they can mature way more and for way longer. So probably I would say those could be actually, the transplanted brain organoids could be an amazing model for studying aging. And I know that people do use them. The only thing that you really need to be careful of is they have obviously a limit that is given by the fact that they're lacking, for example, again, blood vessels, so the media can't pass through the organoid that easily. And they're lacking, for example, microglia, which has been shown to be extremely important in aging and in general, like in neurodegeneration also. And so, yeah, I think probably the transplantation might be like actually a good model for aging.
And So yes, we can provide some references. And there's another question regarding catalog numbers and technical issues and protocols. I think with the references, we can definitely provide you with that as well. So we'll take a final question. How many cell types for the brain organoids are used?
How many cell types? As if inside the brain organoid, or?
I don't know, there's billions, I think 10 billion neurons in the brain, human brain anyway.
Yeah, so if it means how many cell types there are used for making the brain organoids, usually they're either iPSCs or human embryonic stem cells. If you mean inside the brain organoid, there are really like different, as I show you, like, and again, that's the power of the brain organoid, right? This cellular heterogeneity that really recapitulates brain development. So there are many subtypes. The way we study them, it's still very limited because we're still trying to understand them. So even the way we subtype probably is not sufficient. And people still subdivide them in morphologically, cellularly, transcriptionally. I think nowadays we're getting also at the protein level to divide them. So there are a lot. There are definitely a lot.
Since it's related, I think we can take this last question. Could we develop organoids from iPSCs derived from blood rather than fibroblasts?
Yes, as far as you can make, you can reprogram them. I think actually people have been done not only from fibroblasts. I think fibroblasts is obviously the biggest example of how to make induced pluripotent stem cells, but people have been making induced pluripotent stem cells also from urine samples, for example. So ideally, as far as you can make an induced pluripotent stem cells that bears that pluripotency potential, yes, absolutely.
And the last question, I can find this, or can I find this type of data to study the cerebellum or cerebellocortex? I think that depends on the differentiation media and the origin.
Yeah, there are some, I think that they try to recapitulate cerebellum. Again, as far as you usually type, there are like, I think nowadays there are a lot. And there are really like many protocols out there. So it really depends on what you want to do, yeah.
Okay, we'll take a final question. Are there severe defects on mice following brain organoid transplantation?
As far as I know, no. So I think it's very preliminary. I think the last 2 years, the main studies about full brain organ transplantation happened. They didn't seem to have much defects. As far as I know, they don't behave differently. They seem to be fine because they literally incorporate. And something that is very fascinating is that, for example, the microglia can migrate inside the brain organoids. So the mouse microglia can migrate, so they actually incorporate. But not if you have added human microglia. So if you have human microglia, in that case, the mouse microglia stays outside. But in general, they tend to incorporate quite well to the limits of obviously having a transplantation. I know that so far the main issues are the efficiency, because obviously it's a tedious process at the moment. But apart from that, as far as I know, yes, they are relatively fine, so no severe effects.
Yeah, there doesn't seem to be any effects or negative effects when you transplant from the organoid to the mice, but a recent study in Science actually showed the other way around. When they took and they transplanted from dogs into then a human organoid, there were severe defects and tremendous delays. The electrophys was completely different. I think it's very interesting. So it seems to go in one direction, but not the other. There's definitely species-specific differences. Absolutely. Okay, that's all the time we have for questions today. I'm so happy you were all so engaged, but I think that's because of Veronica's beautiful talk and Eugenia's as well. So I'd like to thank them both again. If you have any further questions or inquiries, please feel free to contact us. We are social. Check out our social media sites, LinkedIn, our website, our blogs. And I will ask you, reminder, please don't forget to take the survey. It's really important for us, and it will show up in your browser directly when the webinar ends. To address a number of questions that were in the chat function, this webinar has been recorded, and you'll be able to find the video soon on our website, so we don't have to send you a PDF. You will have the full recording of all the slides on our website shortly. And finally, all of us at Atlas Antibodies would like to thank you, Veronica and Eugenia, and all of you, not only for your attention, but for joining us today, where our antibodies are made by researchers for researchers. So happy staining, everybody.
Thank you.